Hi, welcome to show new channel. Please like, share and subscribe. My today topic is about DNA. So DNA is a deoxyribonucleic acid which is hereditary material in all the organism and is responsible for carrying hereditary characteristic feature from one generation to another. The information stored in the DNA is in the form of code made up of 4 chemical bases. These 4 chemical bases are A, C, G, T, G. So A is adenine, G is guanine, C is cytosine and T is thymine. So you can remember this from by alphabetical also, because it will be very easy for you to remember A, C, G, T. So how they look? So this is the adenine structure, this is cytosine structure, this is guanine structure and this is thymine. So after completing this, now how it is related to the DNA, how they form code. So the order or sequence of these bases determine the information available for building and maintaining an organism. And these codes are known as base pair, which are attached to a sugar molecule and a phosphate molecule. We will see this how sugar molecules and phosphate molecule later in this video, and they pair up to form 2 base pair which are arranged in 2 long strands in the form of ladder, a spiral structure called double helix, and simply they are rearranged to create life from bacteria to human. Remember, to create life from bacteria to human. So now you will understand why I have told you bacteria. This is a diagram of a single cell bacterium. The scientist of the Scripps Research Institute (TSRI) has discovered the first stable semi-synthetic organism, that is a single cell bacterium, that may be important for the discovery of drugs or medicine and other applications. So but do you have any idea what is semi-synthetic organism? I will explain you. It is an organism that relies on a man-made part, to function as a part of its essential biology. So the scientists of TSRI created a new bacterium that has all 4 natural bases. We have learned A, T, C, G, and which every living organism have, but also have 2 pair of synthetic base X and Y in its genetic code. And it is said that if the semi-synthetic organism is really an organism, then it will stably maintaining the information by dropping X and Y base pair, as the additional information was used by the organism possessed in their DNA. So in this we have to drop X and Y. So after this, for this they have optimized a tool known as Nucleotide Transporter, which bring the necessary material for the unnatural base pair copied across the cell membrane. With the help of this, it becomes easier for the organism to grow and divide while holding on to X and Y. Then copying their synthetic base pair becomes easy for the scientist by optimizing the previous version of Y during DNA replication, creating different molecules that can be better recognized by the enzymes that synthesizes DNA molecule. So you have heard that in this we have to drop X and Y, and this is by this you can do lot of means we are going to use as how for getting the information. So later on the scientist has developed one more tool, which is a gene editing tool, which is CRISPR-Cas9 is used by the researcher in such a way to see a genetic sequence in an organism without X and Y as a foreign invader. With the help of this, a cell that dropped X and Y would be marked for destruction and can be held for a new basis by the scientist. Amazing is that the semi-synthetic organism was able to keep X and Y in its genome after dividing 60 times. 60 times, which makes scientists believe that it can hold on to the base pair indefinitely. So what is CRISPR-Cas9? It is a unique technology that enables geneticists and medical researchers to edit part of the genome by removing, adding or altering section of the DNA sequence. You can see this is how it is, how by this diagram you can understand how this is used to know the target sequences. So if you want to know this DNA, you can do this. So this is it is currently the simplest and most versatile and precise method of genetic manipulation, and is therefore causing a buzz in the science world. So after knowing the DNA and its tools, we will see the structure of DNA. And when we say the structure of DNA, the two names comes in front of us is James Watson and Francis Crick. So in 1953 they have discovered the structure of DNA and told that the function of DNA depends upon its structure only. So whatever you are seeing, the information, it's all depend upon its structure. So these are a picture of James Watson and Francis Crick. So this is the structure of a DNA, which is, you can see the double helix, and you can see this adenine, thymine, guanine and cytosine, and here in this A, G, T. So let's see this. First of all, we have seen this is a long chain, and long chain of polymer made from from repeating unit called nucleotide. So this adenine, thymine, guanine, cytosine are nucleotide monomers. So these 4 nucleotides are made by sticking of phosphate group and nuclear base to a sugar. This sugar is called deoxyribose. In this figure you are not able to see this deoxyribose and phosphate group, but in the next figure you will definitely going to know how this whole structure of DNA has formed. Let's see this one more point. It is present in the form of ring. You can see this is all present in the form of ring containing 1 oxygen and 4 carbon, and also contain hydroxyl group. This hydroxyl group OH attached to the 3rd carbon in a ring. So by this figure now you can see phosphate group and deoxyribose and nitrogenous base, 3 and 5 and everything, and even hydrogen bond, hydroxyl group. So let's see this more. In DNA polymer, phosphodiester bond that join one DNA nucleotide to another are always linked 5th carbon of the first nucleotide to the 3rd carbon of the second nucleotide, and as a result a covalent bond is formed. You can see the covalent bond is formed. Now chromosomal DNA consists of 2 polymer of DNA, which made a 3D structure called a double helix. You have seen the diagram of DNA. It is a double helix, and in this structure strand of DNA run anti-parallel. You have also seen this structure was anti-parallel. That is, 5th end of one DNA strand is parallel with the 3rd strand of other DNA, distinguishing the 3rd and the 5th end of a DNA strand. A strand of a DNA consists of a linear polymer of a DNA nucleotide. The backbone of the DNA strand consists of a repeating pattern of sugar and phosphate group, in which the phosphate of one nucleotide is covalently attached to the sugar of the next nucleotide. This sugar phosphate group arrangement is called phosphodiester linkage. I am talking about this. This is a phosphodiester linkage. The 2 end of of a DNA strand are distinct from each other. The 5' has a phosphate group which is attached to the deoxyribose. So you can see this is deoxyribose sugar, and this is at the end there is a hydroxyl group OH attached to the 3'. You can see in this is attached to the 3', and each DNA strand formed by nucleotide is connected by a non-covalent bond called hydrogen bond. So you can see here a hydrogen bond is formed. One interesting thing is this: you can see the pair of this bonding. A is bond with T, C is bond with G. See how they have formed a bond. A and T each have one donor and one acceptor, while in case of cytosine and guanine, cytosine has one donor and two acceptor, while guanine has one acceptor and two donor pair with each other. So that's why you can see here 2 and here 3 bonding. This is how it formed, and the whole structure are bond like this. So, and these are continuous chain, and this is a long chain. And after understanding the structure of DNA, now it's time to understand its function. So polymer of DNA direct the production of other polymers called protein. Okay, so a chromosome consists of smaller segment called genes, and each gene is divided into 3 nucleotide subsegment called codons. Okay, so from DNA to a chromosome, chromosome to genes, and genes is again subdivided into a codons. So from whole of this lecture you understand that DNA is the information molecules which stores instruction to form a large molecules called protein, and these instructions are stored in each cell and distributed among 46 long structures. These 46 long structures are called as are nothing but chromosomes. These chromosomes are made up of small segment of DNA called genes. The transmission of genes to an organism offspring is the basis of the inheritance of phenotypic trait, or it form organism which is like a parent organism, like a man give a man to a bird, lion to a lion, and so on. The genes also regulate the growth of an organism, that is from puberty to adulthood to old age and death. It is also responsible to a specific character to the individual, like the color of the eye, the color of skin, and determination of the sex, and many things. So thank you a lot for watching this this lecture. Thank you.